Hi and Assalamualaikum, welcome to channel 8. For this assignment, we were given an article by researcher King Tang and his colleagues regarding Cyclic-di-GMP or c-di-GMP contributes to adaptation and virulence of Bacillus thuringiensis through a riboswitch regulated collagen adhesion protein or Cap. What we learned from this article is the importance of c-di-GMP riboswitch and its effect on some biological and phenotypic of Bacillus thuringiensis. So in this manuscript, they found that the expression of complete Cap gene in the parent strain to be 10 times lower than in the mutant strain of Bc2 RNA, which represents the repression of Cap in the parent strain. In this experiment, they studied and compared the observation between these two strains to observe their motility, exopolysaccharide or EPS secretion, cell aggregation, biofilm formation, and even the virulence into host insect larvae. So, these results and observations were achieved by 13 different methods, such as virulence assay and aggregation assay.
Based on their study, there are many important results that could help us understand about c-di-GMP. The first observation is Bc2 RNA is located in the 5' untranslated region of Cap mRNA. Bc2 RNA contains terminator sequence which regulate the Cap expression. This result were obtained by doing multiple sequence alignment with Bc2 RNA which have been studied to regulate gene expression. This observation is further analyzed by bioinformatics analysis. And from the analysis, there are 228 nucleotides from the transcription start site to the Cap, and the Bc2 RNA is in the region from 151 to 197. The second observation is Bc2 RNA repress the Cap gene expression. They compare the expression between a complete Cap gene with a Bc2 RNA and a Cap gene with deletion mutant of Bc2 RNA. The results show a lower amount of Cap expression in the complete gene than the mutant gene. This indicates Bc2 repressed Cap expression. Next, the third observation is the binding of c-di-GMP to Bc2 RNA enhanced Cap transcription either in vitro or in vivo. Both in vitro and in vivo observation indicate that Bc2 RNA repress Cap expression, and the binding of c-di-GMP can lead to de-repression of Cap. The fourth observation is that enhanced Cap expression can inhibit motility and block EPS of b.thuringiensis. From their observation, Bc2 mutant show the least swimming motility and EPS secretion due to the lack of Bc2 RNA which promote the Cap expression.
Next, King Tang also found that bacterial motility and EPS formation are related with aggregation. They stated that both Bacillus thuringiensis and Bc2 RNA-regulated deletion mutant efficient in enhancing cell aggregations. However, in Cap deletion mutant, it is remained because no Cap gene can do the transcription process. Besides, biofilm formation in bacteria can be declining as the Cap expressed excessively. Bacterial biofilms are communities of cells that bind together by cell-produced extracellular matrix, which give mechanical stability. The result found that the absence of Cap in Cap deletion mutant make the biofilm to be thicker. However, in Bc2 RNA-regulated deletion mutant, the biofilm is thin because Bc2 RNA is absent to repress the Cap deletion mutant. For your information, b.thuringiensis produces virulence factor and biofilm formation is released with it. Based on the result, Cap will express excessively in Bc2 RNA-regulated deletion mutant and exhibited stronger insecticidal activity because it abolished the formations of biofilm. From the result, we can discuss that when c-di-GMP is in lower concentration, Bc2 RNA-regulated transcriptions of Cap by de-repression or de-repression model. However, it is still allows some leaky Cap to maintain their physiological function. So, this Cap can lead to lower motility and EPS secretions, lower biofilm formation, and higher aggregation rate.
So, after we know about King Tang and colleague research, what makes their work important? Today, there are problems about the pest problem for the farmers to grow their crops. So, there are solutions. In their works, they found that the Cap and biofilm affect the virulence of Bacillus thuringiensis. This virulence may affect the insect larva by produce the intracellular insecticidal crystal protein (ICP) that kill wide variety of insect larvae. This finding made an interesting foundation on creating a biological pesticide which can help farmers to grow their crops. This can be seen in the quintang and colic dishes by using virulence assay system of Helicoverpa armigera as the host. Helicoverpa armigera is a moth, the larva of which feed on the wide range of plants, including many important cultivated crops. The results show that the growth of these hosts are correlated with cap and biofilm. The mutant Bc2 have the highest insecticidal activity that show the ungrowth of Helicoverpa armigera. So, due to the combined virulence effect of cap protein and biofilm formation, we can create pesticide with this bacteria. So, let's get inside.
From this article, I found that the importance of this article is to reduce the pollution by using biological pesticide. Back in the days, farmers used more toxic pesticide rather than using this new kind of technology. There are 3 types of biological pesticide, which is plant incorporated, chemical and microbial. The one we are using is the microbial one, which we are using the Bacillus thuringiensis, genetically engineered. Farmers nowadays are more open to use this kind of technology because they know dangerous effect that will affect the environment. The gene that is responsible for causing intercellular crystal protein that we found in this article, I ran the gene in the blood and found almost similar bacteria, which is the strain YWC2-8 and another strain which is Cerovacastogi strain. It also uses a very specific and sensitive method to obtain a very delicate result. For example, B-galactosidase assay and in vitro transcription termination assay.
So this marks the end of our video. Thank you for watching. Bye!